Good day to everyone joining us, and welcome to today's XTalks webinar, Spray-Dried Dispersions, Robust Formulation and Science of Scale from Preclinical to Launch. My name is Seti Abdo, and I'll be your host for today. Today's presentations will run for approximately 60 minutes, followed by a Q&A session. This webinar is designed to be interactive, so please feel free to send in your text questions and any comments that you have throughout the presentation. You can do so by using the questions function that's located in the panel on the right-hand side of your screens. The presentation slides will advance automatically, and if you need any further assistance, please contact me at any time during the presentation by sending a text message in the same questions panel. We'd now like to take a moment to thank Bend Research, who have helped develop the content and presentation for today's meeting. For more than 35 years, Bend Research has worked with clients to create value by advancing new medicines that improve human health and to solve their most difficult scientific and technical problems. This success is based on the company's ability to develop, advance, and commercialize pharmaceutical technologies. The firm's innovative drug delivery solutions grow from a solid base of scientific and engineering fundamental understanding. Bend Research has more than 200 employees based in 4 state-of-the-art facilities in Bend, Oregon. For more information about Bend Research, please contact Phoenix Ivers. You can see her email and telephone number on the screen in front of you. We'd now like to welcome our speakers for today's conference, Dr. Corey Bloom, who's the Director of Formulation Science, and Dana Settell, who's the Director of Engineering Late-Stage Programs at Bend Research. Dr. Bloom is the Director of Formulation Science and has been with the company since 2003. Dr. Bloom's responsibilities include leading a team that develops pharmaceutical formulations to improve stability and bioavailability for oral and local drug delivery, designs in vitro experiments to assess new dosage forms and formulations, and develops biological models to predict drug absorption, distribution, metabolism, and elimination. Before being named Director of Formulation Science, Dr. Bloom held the positions of Group Leader and Senior Research Chemist. He holds a bachelor's degree in chemistry from Gustavus Adolphus College in St. Peter, Minnesota, and earned his PhD in Analytical Materials Chemistry from Colorado State University. Also joining us today is Dana Settell. Ms. Settell is the Director of Engineering at Bend Research and has been with the company since 1999. She is the former Director for Operations at the company's current Good Manufacturing Practice facility, where she assisted with the installation, commissioning, startup, maintenance, validation, and operation of a PSD2 FSD4 spray dryer. In addition, she oversaw all aspects of production at the facility, which manufactures drug products that met CGMP standards. Ms. Settell has extensive experience with scaling up drug production from development through commercial production, and she was instrumental in technology development, technology transfer, and the startup of $90 million commercial plant in Ireland to manufacture our spray-dried dispersions, technology at the metric ton scale. Ms. Settell holds a bachelor's degree in chemical engineering from the University of Colorado, and she holds 2 patents and is a member of the American Institute of Chemical Engineers. Participants can access more information about our speakers and sponsors at the event's main page on www.xtalks.com. And now, without further ado, I'd like to hand the mic over to Dr. Corey Bloom. You may begin when ready.
Okay, thank you for that introduction, Seti. I am Corey Bloom, as you said, and Dana Settell is here with me and work from Bend Research. And again, we'll be talking about spray-dried dispersions, robust formulations, and science of scale from preclinical to launch. That nice picture there is a picture of the Cascade Mountains, which are just to the west of us here in our facilities in Bend, Oregon. We'll have just a couple of quick slides here about who we are and what we do at Bend Research, and then we'll get into the meat of the talk. So just briefly, it's perhaps not obvious to everybody where in the world Bend, Oregon is, and so here's a little map that shows we're just about right in the middle of the state in central Oregon, which is just on the eastern side of the Cascade Mountains. Our company has about 225 employees at present, and we have 3 main facilities, which are shown here, that span from research and development and analytical. Here at our Tumalo facility, and a little bit more in town, all next to each other, we have engineering and scale-up facility, and our main GMP manufacturing facility. We also have a pilot GMP manufacturing facility where we do low bioburden work for spray drying for applications such as pulmonary. And we also have a new facility that we're expanding into to increase our GMP manufacturer capacity. As far as the technologies that we practice, we are in the drug delivery business, and we have a number of what we call mature technologies that revolve around oral delivery, both for solubilization of poorly soluble molecules as well as controlled release. We'll focus today, of course, mostly on spray-dried dispersions, so we really won't touch on a lot of the other things that are on this slide outside of this brief mention here. So we'll be talking again mostly about spray-dried dispersions, although we do have some other solubilization technologies that we practice, such as hot melt extrusion. And we'll talk just briefly about inclusion of SDDs in those solid dosage forms. We have a number of other technologies we call emerging, in that they have not yet been to the clinic, but we are progressing towards that. And most notably, that's in pulmonary delivery or inhalation, which is a pretty straightforward extension of our expertise in spray drying and particle engineering. So if you have questions about those topics, certainly we'd be happy to discuss them in a venue other than the webinar today. So with that, we'll move again to the main part of the talk. And this is just an outline to sort of orient us. So start out with a fairly brief introduction to spray-dried dispersions and what they are and why we would use them. Then talk about selection of a robust SDD formulation, kind of focusing on the three key attributes of performance, stability, and manufacture. Touch briefly on the inclusion of SDDs in the solid dosage forms. And then Dana will speak much more extensively about manufacture and scale-up of SDDs, as well as a quality by design approach. Let's move into sort of the introductory material on spray-dried dispersions. This is kind of a real high-level overview of the process and the characteristics of spray-dried dispersions. So on the left here, at the highest level, what's done is the drug and polymer are dissolved, or sometimes polymers are dissolved in organic solvents or a mixture of solvents, and they're spray-dried into an expansion chamber, and the powder is collected. Dana's going to talk much more about that, so I'll leave it at that for now. But when done properly, what's achieved is a high surface area particulate with typically a morphology, kind of what's shown here in this SEM image. So you have kind of a collapsed sphere or a spherical morphology. And again, when done right, you have a homogeneous, stable, amorphous dispersion, which can be evidenced by thermal analysis like differential scanning calorimetry to have a single glass transition temperature. No evidence of crystallinity by powder X-ray. Again, when executed with the right polymer, the right drug-polymer ratio, and the proper manufacturing, what the advantages give you for poorly soluble drugs is rapid dissolution, increased solubility relative to crystalline forms, and maintain supersaturation or maintaining that increased solubility in the intestinal fluid. And so as such, you get increased absorption of a poorly soluble drug. And then SDDs can be included in a number of dosage forms, which we'll discuss more in a few slides. Okay, so this just kind of highlights a little bit of the performance advantage and experience with spray-dried dispersions. They've been used extensively by ourselves and others, both in preclinical and clinical studies to provide greatly increased exposure relative to crystalline drug or other dosage forms. And there's some examples shown here of clinical data as far as late phase 3, showing the increase in AUC for SDD relative to even a soft gel or crystalline comparators, and then just a few examples of preclinical species. So the technology is pretty well precedented for a pretty broad array of drugs from chemical space. And there's a simplified map of chemical space that sort of illustrates that here. I'm going to touch on that more here on this slide, so we'll move on. One of the ways that we can leverage our fundamental understanding of spray-dried dispersions and also our experience with them is through the use of a number of kind of maps like this. So this is a simplified map of drug chemical space, and this is one that's been published in the literature. The paper was authored by a number of current and former Bend Research and Pfizer employees. so you can find this paper in the reference that's down at the bottom of the slide. But what it is again a simplified map of chemical space that aids in the selection of drug polymer ratio. It was particularly built around the HPMCAS polymer, which I'll discuss further. Although it does have utility more broadly, in fact, as well. But the axes on this map are on the y-axis you have ratio of melting point over glass transition temperature, or TM over TG, which is effectively the driving force to crystallize. And then the x-axis is lipophilicity, or log P. So the compounds in here kind of span from in the upper left-hand corner of the plot here. You have your brick dust, or your very highly crystalline compounds that are difficult to dissolve in anything. Very high melting point, and do have a strong driving force to crystallize. So the challenge for those, particularly in an amorphous form, is to maintain that amorphous state and prevent recrystallization. And so as such, oftentimes what's done in formulation, if you formulate these as a dispersion, is to go to fairly low drug loading, typically in 10 to 25% drug loading in the dispersion. And in the other corner of the plot, you have the very greasy molecules, very high lipophilicity, low crystalline forces, and in fact, in the extreme, some of these are oils at room temperature. And the challenge there is not one of preventing crystallization, but rather getting rapid and extensive dissolution for something that's so hydrophobic, even when it's in a dispersion. So there again, sometimes you go to fairly low drug loading in the SDD, or in fact, there are a number of related dispersion technologies to increase surface area and dissolution rate that we sometimes use. And then in the other parts of the map, you get into kind of more middle-of-the-road compounds in terms of lipophilicity and crystalline forces, and there oftentimes you can increase the drug loading to 25, 50% and still have good performance and stability. So we use maps like this and a number of others that are called out on the bottom left. I won't read them off, but the point is you can leverage fundamental understanding of the science behind dispersions as well as experience, empirical experience in formulating to screen a fairly small number of formulations to select one that's going to give you acceptable performance and stability, rather than screening a very large number. So that's a good way to proceed as kind of a paper exercise before beginning formulation. As I mentioned, we do use extensively, but not exclusively, the HPMCAS polymer. So this kind of just describes that polymer a little bit and why we use it a lot. So HPMCAS is hydroxypropyl methyl cellulose acetate succinate. And so if you're not familiar with it, the structure is shown here. It is a modified HPMC, basically, if you're more familiar with that polymer. And it is an enteric polymer in that when the succinic acid groups are protonated, it's insoluble. So in the stomach, it's insoluble at low pH. And then at higher pH in the intestinal media, when the succinic acid groups deprotonate, it becomes soluble. It comes in 3 grades that have different acetate and succinate contents, as shown here. And then as such, they dissolve at different pHs. And the polymer is commercially available from Shinetsu in Japan, and it is precedented and approved for use in the U.S., Europe, and Japan. So some of the advantages of this polymer, as it says, one in terms of performance, is with the large number, with the amorphilicity and the large number of kind of chemical groups, there are a lot of handles, if you will, for interaction with drug molecules, and it tends to perform particularly well with respect to maintenance of supersaturation in the intestine, and we'll kind of look at a model for that here in the next slide. With respect to physical stability, it has a number of advantages too, in that it has a fairly high glass transition temperature in general, and particularly it maintains that high glass transition temperature at high humidity, so that because it doesn't take up water, it isn't nearly as hygroscopic as a polymer like, say, PDPA or some of the other dispersion polymers that are used. You can maintain a fairly high glass transition temperature at high humidity, and as such that provides a lot of benefit for physical stability in the solid state, and we'll again kind of return to that, to the rationale for that and why that's important shortly. Just to be clear, though, there are certainly other polymers that can and that we and others do use for SDDs. And so it's not to say that HPMCAS is always the right one or always the best one, but oftentimes, in fact, it does have significant advantages. So here, this slide kind of describes one model for how HPMCAS SDDs dissolve in the intestinal fluid and how they provide a performance advantage. So what's shown here is that the SDD particle comes in, it begins to hydrate as the polymer is soluble again in the intestinal fluid, and then it dissolves and oftentimes casts off these kind of colloidal drug polymer species, which are shown here. As well, of course, you get rapid dissolution from this high surface area material to the free and dissolved drug, and then that's also in equilibrium with biosalt micelles. So the free drug and the biosalt micelles diffuse across an undisturbed mucous boundary layer in the intestine, where the free drug is absorbed. And then the SDD and the colloidal species can continue to rapidly source that free drug as it's depleted through absorption. And of course, again, the idea is that you have higher solubility for an amorphous form than for the crystalline form. So as such, you have increased free drug relative to what you have for crystals. And importantly, again, when done properly in terms of selection of the polymer and the drug-polymer ratio, you can prevent crystallization back out of that supersaturated solution. And so that's where the the drug-polymer interactions in the solution state become particularly important. And again, often but not always, that's where HPMCAS particularly shines. I should say that's one model for the dissolution performance that we often see, but not always. And so as we're studying the dissolution of SDDs through a number of different methodologies, that's one thing that we always seek to understand is what is the dissolution mechanism? Does it follow the model I just described, or is it more of an erosion model? What are the drug-containing species in solution? And you can use that knowledge to understand how your formulation works and to understand what are the key attributes. So in terms of dissolution, in vitro dissolution performance, this is a model drug example. It's an HPMCAS SDD, as a PDPA-based SDD, as amorphous drug alone, prepared by spray drying without a polymer, and then it's a crystalline drug. And this is a kind of a typical microcentrifuge type of dissolution study in a simulated intestinal fluid. And what you see is what's often seen in that the SDDs both provide rapid dissolution and supersaturation, although the PDP SDD in this case crashed out quite rapidly, whereas the HPMCAS SDD continued to provide that good sustainment that you really want to see. And that is often, but not always the case. So what you do, of course, during your selection of an SDD formulation early in development is select your polymer and your drug-polymer ratio to achieve this kind of performance, rapid dissolution, and maintain supersaturation. Of course, in addition to performance, you also have to consider physical stability, which we'll touch on next. But if, obviously there's a logistical driver typically to maximize drug loading in the SDD, and so you want to, as much as possible, typically increase drug loading while maintaining performance. Now, some of the things you can see as the SDD, as you go through project progression and scale up, in some cases, particularly when the dissolution mechanism is not the one that we described on the previous page, so say if it's more of an erosion type of mechanism, you can start to see, or when you have a particularly hydrophobic drug or hydrophobic dispersion because of high drug loading, you can start to see particle size effects in terms of dissolution rate, and that's kind of demonstrated in a hypothetical case here in this lower plot. So as you go to bigger particle size, in those types of cases, you can start to see slower dissolution, which of course you want to minimize. So this is the type of thing that you want to understand in early development and either formulate around it by lowering the drug loading or changing your polymer if possible, or you need to understand during scale-up and manufacture how to control attributes like particle size. So these kind of studies are helping you, to understand the mechanism, to build a knowledge base, and to help define critical to quality attributes later in development. I think, just real briefly, I'll touch on this slide and just say that in addition to kind of your typical dissolution studies, there are a number of other ways, again, to tease out the speciation of drug, if you will, or what types of drug-containing moieties are present in simulated intestinal or gastric fluid. So you can use things like ultracentrifuge, NMR, et cetera, to interrogate what are the different, when is drug in a precipitate, when is it true dissolved, when is it biosalt, misalt, et cetera. And again, all that understanding can help to understand the mechanism of dissolution and thus understand how to control SDD performance. So performance obviously is key, and then stability is just as important for an amorphous form. So with using a fundamental understanding of science of dispersion stability, and this is pretty well understood at this point, we use what we call kind of a phase-appropriate stability testing cascade. It's kind of spelled out here a little bit. So early in formulation selection, we use kind of a rule of thumb methodology based on glass transition temperature that I'll describe on the next slide. Then as you progress, you can do more rigorous physical stability mapping, as well as real-time accelerated stability studies, which obviously are always key in the end. So we kind of go through this cascade so that as you're advancing a molecule, you understand more and more, but you have a very good idea what to expect from physical stability. This slide kind of describes in early formulation selection development the use of kind of a rule of thumb approach. And there's kind of two ways you can have a physically stable dispersion. One is if you're lucky enough to have a thermodynamically miscible system where the drug and polymer are in a thermodynamic solid solution at the selected ratio. That's most oftentimes not the case, and so in fact we just go on the assumption that is not true. And as such, the dispersion is kinetically stabilized. The mechanism for kinetic stability of a dispersion is that when it's stored well below the glass transition temperature, in fact, the mobility in the dispersion is low enough that you simply don't observe phase separation and nucleation of crystals at any relevant time scale. And so basically what you're doing in this rule of thumb kind of analysis here is you're measuring glass transition temperature as a function of humidity, and then comparing that to some of the key accelerated storage conditions like the ICH condition. So in the example on the left here, you measure glass transition temperature, and this is done using modulated DSD. You measure glass transition temperature as a function of humidity and compare that to storage conditions. In this case, if you look at the most 75% humidity and the most aggressive kind of ICH condition is 40 C, 75% humidity. At 75% humidity, you have a TG of 46 degrees. And so there's only a delta or separation of 6 degrees between the TG and the temperature at this humidity or the storage condition. So you'd really like to see at least, ideally, a 20-degree separation or at least a 10 or 15-degree separation. And so the prediction here would be that you are likely to have high mobility at this condition and ultimately nucleation and crystallization. And in fact, that's exactly what's observed after 3 months. Whereas at other conditions where there's a bigger differential, you're storing it well below the glass transition temperature, and with the lower mobility, you're not going to see the crystallization. So that's also observed. So the thing you would learn from this in this example is that you're not likely to have good physical stability at this condition. And so for this particular SDD, you'd have to package away from humidity. In some cases, you can instead formulate around this. But if you wanted to use this SDD and for this particular drug, you would have to, in fact, package against humidity. Now, in this other example, you'd go through the same exercise of measuring and plotting TG versus RH. You see there's for this particular compound, which is 25% dispersion with HPMCAS-M, there's an 18-degree separation here between the 40-75 condition and the TG at 75% RH. And so as such, we predict that this is likely but not absolutely certain to have good stability at 40 degrees and 75% humidity in an open condition. So you can go through this kind of screening exercise and have a very good idea of where you stand, whether there's very likely to be a problem, where there will not be a problem in any storage condition, or where you're perhaps in a bit of a gray area. So particularly when you're in sort of a gray area, you can kind of go to the next level of more rigorous physical stability mapping using a technique like isothermal calorimetry, or TAM, in which you actually measure the kinetics of crystallization at a given condition. So in this instrument, what you're doing is setting up a sample at a controlled temperature and humidity for many days to weeks, and measuring heat flow, and so you can actually observe the time it takes to reach crystallinity. We typically define it as the time to reach 5% crystallinity as a good metric, although that's admittedly somewhat arbitrary. What's done is you do this at a number of conditions, and you have to know the glass transition temperature through DSC or other measurements. And then you can plot that data as shown below here, where the y-axis is log of the time to crystallize, and the x-axis is TG over T, which is effectively a measure of mobility. And you can plot out the data, which of course is taken at fairly aggressive conditions so that you observe crystallization, and extrapolate that out to less aggressive conditions. And you can predict what are the kinetics of stability over time at various conditions. So in this particular example, the prediction would be at 75% humidity and 40%, 40 degrees C, that you're going to see crystallization, again, in that you have high mobility, but at 40 degrees, 25% humidity, that you will not see it for at least 2 years. And in fact, data shows that where stability is predicted, it's observed. So this is, using this kind of methodology, you can map out solid state stability a little bit better. Here's one more example. This is with actually a 90% active dispersion. So this was a very high TG drug that allowed you to have a much higher loading and still maintain a high glass transition temperature. Same kind of methodology was used to make a map here of physical stability. You can translate that over into a TG, a storage temperature, TG versus RH kind of plot, and predict that in this particular case that you will have 2 years or greater stability at even the most aggressive conditions. So that's the kind of exercise you can go through to be more rigorous beyond the sort of rule of thumb exercise. And finally, just to show all that prediction is nice, but of course this is done in parallel with gathering real-time stability data. The idea is you're pulling those samples and checking off that you're matching your prediction, of course, rather than hoping for the best. And there is plenty of real-time, real-world stability data like what's shown here. I realize it's kind of hard to read, but what's shown is long-term stability data at a number of conditions, including open at 40-75, that shows no physical or chemical, for that matter, changes for this particular SDD, which again was a 25% active dispersion with HPMCAS. So that kind of covers, again, the performance and stability metrics, and of course, again, Dana here very shortly is going to talk about the manufacture attributes of SDDs. Finally, just quickly here, with respect to dosing SDDs, there are a couple options. For immediate release, SDDs are well amenable to suspensions, and they can be nicely used for animal tox or efficacy studies. Early in development, they can also be taken as a powder in a bottle into the clinic, for phase 1 or human PK type of studies. The standard approach that we take for a solid dosage form is immediate release is a tablet, usually using a dry granulation approach that I'll describe on the next slide. They can be placed in a capsules as well, although I would sound just a bit of a note of caution there that when placing them in the capsules, it's oftentimes necessary to do some formulation to provide good disintegration and dissolution and avoid sort of gelling and slow release from the capsule. So it's not always a great idea to just shove the SDD straight into a capsule and dose it. So that's certainly something you want to look at if you go that approach. SDDs can be placed into a number of controlled release presentations, including a soluble core or an osmotic type of tablet, as well as a spray layer dispersion. So with respect to immediate release dosage forms, again, the standard methodology that we use is an immediate release tablet. And the idea here is typically to sort of recover the performance of the native SDD and basically match what you have with the suspension. And so as such, you want a rapidly disintegrating tablet. The approach we usually use is one of dry granulation, using disintegrants and some other excipients. And SDDs are nicely suitable for this, particularly if manufacture is done keeping in mind some of the key particle properties such as particle size, bulk and depth density or flow characteristics, and compressibility. And Dana's going to speak to that in detail here in the manufacturing section. So when manufactured properly, they're quite amenable to a dry granulation approach. In some cases, a direct compression approach is possible as well. And again, what you're seeking to get is rapid disintegration to sort of recover and match the performance of the SDD itself. And there's an example of that shown here on the right. So with that said, I'm going to turn it over here to Dana.
Thanks, Corey. And I'll just finish out the talk here by going over a bit of background on the spray drying process, some methodologies for initial process definition, and some considerations for scale-up within a QBD framework. So moving right into some background on the process, spray drying process. As Corey mentioned, the first step is to prepare a feed solution where you dissolve the API and the polymer in a suitable spray solvent. Some common solvents are acetone, methanol, THF, and blends of those with water, although we have used quite a number of different other solvents as well. That feed solution gets pumped through an atomizer where it gets broken up into droplets that are contacted with hot drying gas. We typically use nitrogen as the drying gas because we're working with flammable solvents. So almost immediately after the droplets are contacted by the hot drying gas, solvent begins to evaporate, and the droplets begin to evaporatively cool such that they're at their wet bulb temperature and will remain at or below the outlet temperature of the spray dryer through the entire drying process. So the spray drying process can be relatively mild temperature-wise, so it's suitable for APIs that may be heat or temperature sensitive. Most of the polymers that we work with are film, for spray dry dispersions, are film forming polymers that form a skin almost immediately as the solvent begins to evaporate. And that skin acts as a barrier or resistance to diffusion of the solvent from the interior of the droplet to the exterior. And then as the drying process proceeds, that skin thickens and hardens until you end up with your final spray dried particle. Of course, the rate or temperatures at which that drying process happens can greatly impact the morphology of the SDD, the resulting SDD, and we'll talk about that on the in a few slides here. The drying process happens relatively quickly. Within seconds, you end up with your dried particle. So we tend to focus on the key control volume up near the atomizer, where the droplets are formed and dry very quickly to define your particle morphology. And that key control volume up near the drying gas disperser is illustrated by a couple of computational fluid dynamics model outputs shown here on the right of this slide, illustrating that point. There's a temperature contour showing that the temperatures within the dryer are mostly at the outlet temperature of the dryer, with the exception of a colder evaporative spray plume up near the drying gas lid. On the right here, we're showing another computational fluid dynamics model output with a user-defined function that models the individual droplet drying so that you can actually trace droplet drying from the wet droplet through the skinned phase where it still may be sticky, and then into the final dried SDD particle. So we've shown here also on this slide a number of the key spray drying process parameters, and there are quite a few of them. So how do you begin to identify your initial process conditions? So moving on to the next slide, we've developed a methodology for doing that initial process identification. Over the years at Bend Research, we've been lucky enough to have developed spray drying processes for hundreds of SDDs. And based on that experience, we wanted to be a little bit more rigorous about initial process identification. So we developed this flowchart methodology, which is a series of boxes that are offline models or tools that can be applied. And then the order in which they're applied helps to minimize the amount of API and development time that may be required to arrive at an initial process identification. And then at the end of this flowchart methodology, you end up with higher confidence in the robustness of the process that you've identified, and also you begin to build the knowledge base that will be useful for future design space work. So we won't go through each of these boxes just due to the time, but comment on a few. Of course, the first step is to identify a robust formulation, and Corey did a good job of talking about through some of those parameters. So ideally, you come into this process identification flowchart with a formulation that is robust enough that the spray drying process parameters have little to no effect on the resulting physical stability of the SDD. In terms of the rest of the boxes, they can be kind of broken down into thermodynamics or spray drying, or thermodynamic or drying kinetics type of parameters, and then droplet formation or atomization parameters. So we'll go through just a couple of those to illustrate the ideas or concepts. So the first box, or the first tool that we're showing here, is just a simple mass and energy balance on the spray dryer itself, functionally relating the key thermodynamic parameters through those simple mass and energy balances. The energy source is supplied by the drying gas stream, and you can think of the energy sink as the heat of vaporization required to vaporize the spray solvent. So on the left here, we're showing a graphical representation of the thermodynamic model with 4 key parameters plotted. The inlet temperature of the drying gas, the spray solution feed rate, and the drying gas flow rate. And these 2 parameters are ratioed to enable us to look across multiple scales on the same plot. It's fair to say that for a single dryer scale, the drying gas flow rate is held relatively constant. So you can think of this axis as your spray solution feed rate axis. Then within the plot, we're showing contours of constant outlet temperature, and also another parameter, another calculated parameter, the lines of constant relative solvent saturation or wetness within the spray dryer. So as you move back to this blue corner in the back here, this ends up being your colder, wetter drying conditions. And you can start to bound the space, the parameter space that you may be interested in running at. So we'll go through a few of those. You can look at a maximum inlet temperature constraint where it could be as simple as an equipment constraint. Your heater simply can't operate above a certain temperature. Or you may want to keep that below the browning temperature of the spray dry dispersion you're working with. And that could be identified with offline hot bench experiments, such that if there are any deposits within the dryer, you know that all of the metal is held at temperatures that are below the browning temperature of the spray dryer. Next, you can look at a process efficiency constraint. So at some point, if the drying or if the solution feed rate drops too low, the spray drying process might just not be viable to progress to the next stage. So usually you have kind of a lower limit on how low you're willing to accept in terms of feed rate. Another constraint is a relative solvent saturation constraint. So you think of this as the dryer can only, you want to operate above a certain relative solvent saturation, or you lose the driving force for drying. So if you get too wet solvent conditions, you won't actually dry. You'll do what we call spray painting, and the material will stick within the spray dryer, and your yields will drop off. So this constraint can be evaluated offline by looking at the TG of the SDD at different exposure conditions to different relative solvent saturations. And the last constraint you may want to consider is an outlet temperature constraint. So you may be working with a material that has thermal stability issues, so you may want to keep your outlet temperature below a certain temperature, or you may have some previous experience or knowledge about a certain type of SDD, and in that case you might understand something about how the outlet temperature correlates with SDD morphology or SDD attributes such as bulk density. So we'll talk a little bit more about that on the next slide. As I mentioned, in the simple case of a single pass spray dryer, the outlet temperature correlates very well with the bulk specific volume or density. We recall that we're working with film forming polymers that as the droplets begin to dry, they form a skin which acts as a barrier to solvent diffusion from the interior of the particle to the outside. And we'll show a couple of high-speed camera movie clips showing a spray solution droplet suspended on a thermocouple and then exposed to different drying temperatures or outlet conditions. So on the right here we're showing a hot outlet temperature which results in hot fast drying, where the skin sets up almost immediately following the almost immediately as the solvent evaporates and acts as a barrier to solvent evaporation, such that the solvent actually evaporates inside the skin and the pressure inside the droplet is higher than the pressure outside, and you end up with these hollow spherical particle morphology that are very compressible. The converse is true for the cold outlet drying conditions, where the skin, the solvent is able to diffuse through the polymer skin at an adequate rate, and you end up with these raisin-like high-density particles that have a little bit less compressibility. Of course, for any drying condition, what you really get is a distribution of particle types, but the process is very amenable to tuning so that you end up with more of one type of particle based on the thermodynamic conditions. And of course, these types of morphologies obviously affect downstream tablet manufacturing, and we'll talk about that a bit more later on. So switching gears and looking at the droplet formation or atomization parameters, shown here on this slide is a pressure swirl nozzle that we work with quite a bit for these spray-dried dispersions. This is just one option for atomization in spray drying. You can use other atomization types like two-fluid nozzles, rotary atomizers, or just the straight pressure nozzles. We find that the pressure nozzles have been very easily tunable and scalable, so we work with these quite a bit. For the case of this pressure swirl nozzle, droplet size is related to parameters such as nozzle geometry, so the size and shape of your orifice and swirl chamber. Also related to nozzle operating parameters like your nozzle pressure, your solution feed rate. And then finally, also related to the properties of your spray solution, probably the dominant property that affects droplet size is viscosity. And these atomization, the droplet size is very difficult to model from first principles, since as the liquid exits the nozzle orifice here, it forms a liquid sheet that breaks up into ligaments and then finally into droplets. And this whole process is heavily influenced by things like turbulence, which is just inherently very difficult to model. But there is quite a bit of literature data out there relating the droplet size to these parameters. So there's quite a bit of existing literature data on those types of correlations, particularly for in the automotive industry. They tend to use quite a bit of these pressure swirl nozzles. So at Ben Research, we we actually measure the droplet size directly using a phase Doppler particle size analyzer installed on an inverted chamber that's designed to prevent drying. So we're really just going in and measuring the droplet size directly. And we can oftentimes do this with the placebo solution where we match the viscosity of the active solution. So a lot of times early on we can just select a nozzle directly by measuring these different droplet sizes under different conditions. Of course, this also begins to lay the groundwork for future correlations between things like nozzle geometry, nozzle pressure, and the solution properties like viscosity. So kind of coming back to the initial process development flowchart, we talked through a couple of the different tools, experimental tools and models on this chart. And I hope that gives you a feel for the kind of knowledge base that you can build on from early process identification. And you can also use these same tools as we begin to scale up and move forward. So talking about scale up, the next slide shows the spray drying equipment located here at Ben Research and the photographs all the way from multiple mini scale spray dryers that we use primarily just for feasibility screening into lab scale spray dryers where you can begin preclinical process development and also do some clinical work for, say, first-in-man type studies. And then several NIRO PSD1 scale spray dryers, and then also an oversized NIRO PSD2 scale spray dryer. But we do also have experience scaling up to larger scale spray dryers, such as a NIRO PSD5. I think the key point here is that you can make commercially relevant or commercial-like SDD at all of these scales down to, say, the lab scale spray dryer. And you can make the same SDD across a wide range of spray dryer scales. So because of that, for design space, we like to take a scale-independent approach to offer the greatest flexibility for post-approval changes. Of course, as you get to the larger and larger spray dryers, it does become easier to make the larger, more dense particles. So you can also take advantage of that if necessary. In terms of a general quality by design approach, we really strive to drive towards that functional relationship between the critical to quality attributes and the critical to quality parameters. And of course, the first choice, or where we strive to first, is a first principles approach. If that can't be achieved, then we would move on to semi-empirical correlations, and then if those two can't be achieved, we'd move on to purely empirical approaches. And of course, the goal is to relate your spray drying process parameters to your key or critical to quality SDD attributes, and ultimately to your downstream or final product critical to quality attributes. And we'll talk a little bit about that on the next slide. So on the next slide, we're showing kind of a general approach for relating your spray drying process parameters to your critical to quality attributes. We like to use these fundamental models to guide experimental design space rather than relying on purely statistical or DOE approach that may result in either nonsensical conditions or conditions that just aren't interesting to study. And then once we've established that, we could use, we relate the spray drying process parameters to the SDD particle attributes, and then from there relate the SDD particle attributes to things like performance and stability or tablet manufacturability. So for instance, Corey mentioned the case where performance can be impacted by particle size. And then in that case, control of the SDD particle size is key, and your atomization parameters become critical to quality parameters. We won't really talk about that case today, but we're happy to go over that if you'd like to contact us in the future. The next few slides will go over a case where SDD compressibility in fact impacted tablet manufacturability. So the thermodynamic spray drying parameters became key SDD parameters. So coming back to the graphical representation of the thermodynamic model, in this case we're adding another dimension to account for another parameter, the condenser temperature, which we need to account for the larger scale spray dryers, which operate in closed loop mode. So the drying gas is actually recycled as it comes out of the chamber. It gets passed through a condenser temperature and then recycled back into the spray dryer. So because of that, it does contain some amount of solvent vapor that obviously affects the drying rate, and that's the particle morphology. So we need to account for that condenser temperature. So we're showing, again, the inlet temperature of the spray dryer, the condenser temperature, again, the solution feed rate and gas feed rate, and contours of constant outlet temperature within the cube here. So the hotter drying conditions are up front and the colder are near the back. So on this next slide, we're showing an example of a 25% HPMCAS SDD experimental conditions where we've plotted PSD1, PSD2, and PSD5 spray drying conditions. And you can see for a very wide range of spray dryer scales, we can operate at similar thermodynamic conditions. This is in this particular case, we span everywhere from kind of on a PSD1 around a 20 kg per hour spray drying or solution feed rate, all the way through to a 500 kg per hour spray solution feed rate. So it's a big difference in scales. Yet they can be operated at similar thermodynamic conditions. And similar to the 2D case that we looked at, you could begin to bound the space that may be interesting to study, or to bound your design space, if you will, by inserting these planes. So you can have, again, a minimum condenser temperature that may be limited by say, utilities or just equipment constraints. Same for your inlet temperature, or you could also limit it to be below your browning temperature. You can also plot within the cube, as we did in the 2D case, contours of constant relative solvent saturation. So the wet conditions or solvent wet conditions are shown back here in the lower or the back right, and then the hot or the drier conditions are shown up front. And again, you can insert a plane, kind of an operating constraint plane, where you don't want to operate the dryer too wet, or you lose your driving force for drying and will begin to spray paint and lose yield. So you don't want to operate in this cold, wet region in the back corner of the cube. So then also kind of bringing in the effect of thermodynamic process parameters on the SDD morphology. In the case of the recycled larger closed loop spray dryers, it's not as simple as just correlating to outlet temperature. So we developed this HMT ratio by performing a dimensional analysis on the heat transfer rate into a droplet over the mass transfer rate out of a droplet, mass transfer rate of solvent out of a droplet. And that resulted in a dimensionless number that we call the HMT ratio that can be evaluated at the readily available process parameters. So the process parameters we've been discussing, the inlet temperature, outlet temperature, gas feed rate and solution feed rate, and condenser temperature. And then you also need to take into account a couple of constants that are specific to the formulation. For high HMT ratios, the numerator is larger than the denominator, so the heat transfer rate into the droplet is faster than the solvent transfer rate out, and you end up with the hollow sphere morphology SDD. And then for the converse is true for the low HMT ratio. That's more like the raisin-like particles. We also developed a mercury intrusion test to quantify the interior void volume of these particles. And it turns out, we won't talk too much about this test just due to time, but it turns out that this test correlates nicely with the tablet manufacturing process parameters. So that worked well for predicting how it might behave in downstream manufacturing. So shown on slide 36, we're showing the HMT ratio plotted against the SDD compressibility or mercury intrusion value. And again, the HMT ratio is evaluated considering all of those key spray drying parameters, thermodynamic spray drying parameters that we've been discussing. So you can think of it a bit as a fingerprint of the spray drying process. So in this particular case, we had a target SDD compressibility that we were trying to achieve, and a target range we were trying to achieve. So if the compressibility or the void volumes got too large, that also correlated with a bulk density, a high or a low bulk density. And you got to a point where you just had flow problems and too much material. The material was too bulky to fit in the bins. So you had kind of an upper limit that you wanted to stay below. And you also had a lower limit in terms of SDD compressibility. And this was just because there was a pre-existing high-throughput roller compaction process, and it was not desirable to make any changes to this roller compaction process. So we had a fairly narrow, tight window that we wanted to stay within. So once you've identified your target SDD compressibility, you can see we've correlated a different HMT ratio for each of the different dryer scales, PSD1, PSD2, and PSD5, and you can read out a target HMT ratio for each of these cases. And remember that HMT ratio then refers back to a combination of spray drying parameters that you can use to achieve that particular SDD compressibility. Might just mention here also, it was interesting to us that there's the difference in the dryer scales, and we wanted to understand that and also take into account the difference in the scales. We won't get into that today just due to time, but we did develop a second dimensionless number that takes into account the differences in the spray drying plume density, which, as it turns out for this case, was pretty extreme as you moved across scales. The spray plume became much more dense at the larger scales and served as a further resistance to heat and mass transfer into and out of the plume itself before it could even get to the droplet. So there are ways to operate the dryers to account for that, and it is also easily predictable. So you can come up with a single parameter that collapses all of the different scales onto the same correlation. So if you bring that information in, you can also start to calculate a constant HMT ratio for all of the different conditions within the thermodynamic space that we've been evaluating. So you can add in a plane of constant HMT ratio that you may want to stay above or below, and remember that relates to the SDD compressibility. you can bound. So once you've put in all these different planes, you can actually bound the space that you're interested in studying experimentally. And within this space, using the HMT correlation, you can calculate the SDD compressibility for any parameter within this space. And although we didn't talk about it today, you could use similar correlations to calculate droplet sizes and thus particle sizes for anywhere within that space as well. So in summary, I hope we've shown today that SDDs are a broad platform for increased solubility and bioavailability. We've shown that SDDs can be used all the way from early development through to commercial scales. The SDD physical stability is predictable and has been demonstrated, and that building a mechanistic understanding of performance and stability facilitates a QBD approach to design space, and that the spray drying process is easily scalable and can be used to control critical and key to quality attributes. So we also just wanted to acknowledge our colleagues at Vendor Search, and then also, of course, colleagues at Pfizer. And a lot of the data we presented today are case studies from Pfizer compounds. So many thanks to the folks at Pfizer. And also thank you for joining us today. And if you'd like to hear any more on this topic, we will be hosting a colloquium at the 2011 AAPS annual meeting. And please feel free to contact Phoenix Ivers for additional details on that or additional questions, and we're happy to answer those.
Thank you very much, both Dana and Corey, for your presentations. We'll now move into the Q&A part of our program, and at this point I'd like to invite our audience to continue sending their questions or comments that they'd like to address to the speakers. You could do so using the questions function that's located on the right-hand side of your screens. I've already received some questions, and so I'll begin with those. First question is, can SDDs be used to improve bioavailability of compounds with permeability, efflux, or metabolism changes? Dr. Corey, perhaps you can answer that.
Sure. Yeah, the short answer is in rare situations, yes, but by and large what we're doing is increasing solubility and dissolution rate. So, you know, there's nothing in the formulation here that directly affects permeability or metabolism. Occasionally, just by increasing the solubility and dissolution, you can somewhat swamp out, say, a metabolism in the gut type of mechanism, or partially overcome poor permeability. But there's nothing direct about these formulations that address those challenges.
Thank you. And another question is, are salt forms amendable to formulation as SDDs?
Yeah, absolutely they are. It's probably more often that we use a free form, either free acid or free base, but absolutely salt forms are amenable to spray dry dispersions. At its simplest, you need something that's going to be soluble in a volatile solvent or solvent mixture like acetone, water, or THF, or a mixture with water. So oftentimes salt forms indeed have perfectly good solubility and in fact sometimes a reasonable TM over TG ratio and can be just fine for spray dry dispersions.
Thank you. And another question is, what polymers are typically used for dispersions other than HPMCAS? And if so, when or why?
Sure. Outside of HPMCAS, HPMC, so the neutral hydroxypropylmethylcellulose, can be a good polymer. Other common ones are PDPA or copovenone, as it's called, or PDP. One reason to sometimes choose those, occasionally there's a chemical stability issue where it's not advantageous to formulate with an acidic polymer, you know, a weakly acid polymer like HPMCAS. So sometimes either for chemical stability or performance reasons, a non-enteric polymer where you have solubility of the polymer in the stomach is preferred. So that's sometimes a reason why you go to HPMC or one of the PVP polymers. The oidragates can also be used, and those also provide high PG in some cases can provide a good interaction, and a different type of polymer chemistry that sometimes provides a good interaction with a few drugs.
Thank you. And another question is, how can you maximize spray dryer capacity, and what impact does this have on the spray dry dispersion product attributes? Dana, perhaps you can answer that one?
Yeah, sure. One of the biggest ways to impact spray dry capacity is by how much solids content you can get into the feed, and that can be limited either by API solubility in the solvent or just the viscosity of the resulting spray solution. So if there's too much polymer in the spray solution, it can actually get too viscous to atomize. So that's kind of a big parameter. And in terms of how does it affect the spray drying process, or the spray drying particle attributes, I would say you can usually tune, with this methodology, you can tune such that you can achieve similar spray dried particle attributes at both the higher capacity and the lower capacity.
Thank you. Another question is, in general, what is your experience for spray-dried dispersions in boosting oral bioavailability over crystalline APIs in suspension?
The experience is very good. We do that very often, for early animal studies, whether it's efficacy talks or what have you. So suspensions of SDDs generally work quite well. You do, when you're screening a formulation, before you would dose in an animal, you absolutely want to do a suspension stability experiment to make sure that once you've prepared an aqueous suspension, that you have a window of time for dosing it. But yeah, we do that very routinely and have had a lot of success in animal models and even, in fact, in human, a powder in a bottle kind of approach for a human PK study.
Thank you. And another question is, what is the relation between dose and TG, and is it the indirect mean to address crystallization?
So I guess by dose, maybe we'll sort of take that as drug loading in the polymer, which is then, of course, related to the dose you can give in a certain unit dose. So that's very much drug and polymer, for that matter, dependent. So the TG, the glass transition temperature of the drug molecule itself is real variable with drug, and in ideal situations, the drug itself has a high TG, say approaching 100 degrees or even higher, and in other cases, the drugs have a very low TG, as low sometimes as 15 or 20 degrees. So when you have a high TG for the drug itself, then that allows you to increase loading of the drug in the SDD and still maintain a fairly high glass transition temperature for the SDD. Where you have a low TG drug, then of course that tends to lower the TG of the dispersion. And then of course all of that's overlaid at humidity. It also depends on the hygroscopicity of both the polymer and drug. So there's a fairly complex interplay there between the drug properties, both the TG of the drug and the hygroscopicity of the drug and the polymer. But I guess more often than not, especially if you're formulating with a cellulosic polymer, the polymer has a higher TG. And so in that somewhat typical instance, then by adding in more drug, you're typically lowering the TG of the dispersion. Again, that's not always the case.
Thank you. And another question is, if successful, how much does SDD technology add to the final cost of a drug?
Yeah, I'll take that one. So typically the SDD cost of goods is very similar to tableting operations. So in general, if you're able to achieve a high throughput SDD process, you can expect the cost to be on the order of a few cents per dose.
Thank you. And is TG in slide 14 of pure drug or dispersion, and what about the TM?
Yeah, that's the TG of the dispersion. So again, you would use differential scanning calorimetry to measure the TG of the dispersion as a function of humidity, and then that's used to determine the TG over. In this case, the T just refers to the temperature being held in the experiment, the TAM experiment. So melting point, in fact, is not directly used in this analysis.
Thank you. And the next question is, could you talk a little bit more about the secondary drying step?
Yeah, sure. We just for time purposes, we left that out. Of course, as the material comes out of the spray dryer, the residence time within the spray dryers are on the order of tens of seconds to a minute. So as the spray dry dispersion comes out, it does have a bit of residual solvent remaining on the SDD particle, and that can be removed through any number of conventional secondary drying options, tray drying, and a method we like to use, a vacuum agitated dryer.
Thank you. And next question would be, what is the impact of spray drying solvent?
Yeah, so there's probably a piece for both of
Yeah,
you. Go ahead.
I guess initially, of course, what you want to do is somewhat maximize throughput, and so you need to have a solvent that you have reasonable solubility of both drug and polymer. It needs to be a fairly volatile solvent or solvent mixture. So the ideal ones are kind of acetone or methanol. THF oftentimes is a solvent that we'll use if we can't get good solubility of the drug and polymer. In acetone or methanol, and then sometimes the addition of a little bit of water can help with solubility. So at the simplest, you need a system where you have reasonable solubility. For throughput purposes, you'd like to approach something on the order of 10 weight % solids in the spray solution. So the sort of upper limit for solubility can be defined by either drug solubility or viscosity. If you have a nicely soluble drug, ultimately the viscosity, which is mostly due to polymer, is the other potential limiting factor. So again, you need a volatile solvent, and the favorites would be acetone, methanol, and THF, at least in our hands. So beyond, I guess, getting a reasonable spray solution, I don't know, Dana, do you want to comment on sort of process?
I think you covered it nicely. We have worked with a number of different solvents. Corey mentioned the common ones, but we've also worked with, ethanol, ethyl acetate, methylene chloride. So there's a number of options out there. And in terms of the spray drying process, as Corey mentioned, it's important to get the maximum amount of material into the spray solution to maximize capacity. But it's also important to consider the heat of vaporization and the boiling point of these solvents, which will also drive the feed rate that you can push through the spray dryers.
Thank you. And perhaps we have a time for one more question. What parameters are most important for preventing spray painting?
Yeah, so as shown in a couple of the slides, slide 22 and so the 3D version of that as well, you can navigate away from the spray painting region by adjusting your other spray drying parameters. So for instance, reducing, say, feed rate or increasing, say, the inlet and outlet temperatures that your process running at. Also, it's important to also note that the spray dry dispersion properties themselves will have a big impact on the propensity for spray painting, and that can be studied offline using various techniques like maybe a TGA analysis of the drop or the spray solution, and then and that can be taken forward into droplet drying models that are also inputted into the computational fluid dynamics models, and we can use those to predict conditions and regions when you might spray paint. Also, another important parameter is the droplet size. So we didn't really cover that part today, but you can get to such a big droplet size that there just isn't time for droplet drying, and you end up spray painting not necessarily for thermodynamic reasons, but more of just a droplet drying and residence time reason.
Well, thank you very much. That is all the time that we have for today. I do realize that many of your questions have not been answered, but they will be sent and to the speakers and the people at Bend Research, and we'll try to get back to you with answers. Again, if you have any more, if you need more information or you'd like to contact Bend Research, you could find the information on your screens. You will also be receiving an email letting you know of when the archive is ready for download, as well as the slides from today's webinar. If you'd like to share those with your colleagues, please reply to that email and let us know. Again, thank you everyone for participating in today's conference, and please join me in thanking our speakers, Dana Settell, who's the Director of Engineering of Late-Stage Programs at Bend Research, as well as Dr. Corey Bloom, who's the Director of Formulation Science. Again, thank you all very much for your time, and we hope that you found this conference informative, and you all have a great day.
The organizer